Hi, this is Dr. Jabali, and in this video we're going to do topic 5.3, which is all about classification. Now in this topic we're going to talk about the binomial system and the names of the species and how it's universal by all the biologists around the world. They agree on how to name them. We're going to talk about the scientific names and how to use the binomial system. We're going to talk about taxonomists and how they classify the species according to the hierarchy of the taxa. We're going to talk about the three domains. We're going to talk about the kingdoms, the phylums, the class, the order, family, and genus, as well as the species. We are going to talk about how the genus, as well as which type of taxa they consist of the species, and what they have in common as between them as ancestors. We're also going to talk about reclassifying some groups because we find out some information about them. We're also going to talk about predicting what kind of species they are by looking at their traits and put them within a certain groups within the taxonomy. We're going to classify one type of plant and one animal species from the domain all the way to the species level. We're going to find features, that means the traits of the different types of phylums of plants, such as Bryophyta, Felicophyta, Coniferophyta, and Aspergillophyta. We're also going to talk about the features, that means the traits that comes from Porifera, Cnidarian, and all the other animals, the other types of animal kingdom phylums. We're also going to talk about the classes of birds, mammals, amphibians, reptiles, and fish. And we're going to talk about how constructing diagram is key to identify certain types of specimens. Now, classification, which is 5.3, it's a lot of information. It's a lot of traits of different types of animals and organisms. Now, to do this one, to actually memorize this stuff, it takes a lot of time. That's why it's important to do different types of map or a project where you're identifying all the traits of all these animals and their names. So 5.3, it can be very long when it comes to memorization. So it's important that it's repetitive, that you see it many times, then you draw or do a project or do a poster in which you're repeating the traits of each one of these organisms in order to be able to memorize for the test. Also, go to Google and look at Google Photos for each one of these organisms, because so many times IB picks up the photos of these animals or these plants and they put it on the IB exam, and you should be able to identify them. So the more you get exposed to them, the stronger you will remember them. So let's start with the binomial system of nomenclature. Basically, this was designed by Carolus Linnaeus in the 18th centuries. And basically what he did was use Latin names. So there's two parts. The first part is the genus and the second part is the species. Now, here's the most important thing that you also need to use when you do your IAs, is that if you're going to do it printed ways, it has to be italics. Now, if you're going to write in the test, and remember, if you're going to write the species and the genus, these two, right here, the genus and the species in a test, you must underline it. If you don't underline, you do not get the points. However, if you're going to type it, let's say in an IA paper, it has to be italics. You don't have to underline. And the first part, which is the genus, has to be capitalized. So here's an example, right? The humans is known as Homo sapiens. You can see from the Homo sapiens that the H, the first one, is capitalized. On the second word, it is not. So, and you can see that the entire word is written in italics. So it's very important that you remember this, especially when in the test. If you decide to write Homo sapien in the test, make sure that you underline Homo sapien. Now, what's the point of this whole hierarchical system of classification? Is the first thing we do is find all their traits. What is that they have in common as traits? And then if they have things in common with them, put them in a similar group. So the more similar they are with their traits, the closer the grouping will be. So there's three reasons, the main objectives why we're using this binomial nomenclature. One is to be sure that each organism has a unique name that cannot be confused with another organism. So we have, let's say, millions of species in the world. None of these species, none of them will have exactly the same name. So that means if a species in China have that name them in Latin, it will be exactly the same name in some other country. So that the names can be universally understood, and no matter the nationality, culture using the name. So like I said, if it's written in Latin anywhere from China to Russia, to Japan, to United States, to Canada, to Africa, anywhere in Africa, these people, when we talk in Latin names, they know exactly what's the species. And so that there is some stability in the system, but not allowing people to change the names of the organism without valid reasons. So all of this purpose of making these binomial nomenclature is just to be able to communicate with scientists all around the world, and for us not to confuse a certain species, because saying a Latin name, we know exactly which kind of organism we're talking about. So here, let's give an example. See, we have two species of sharks that are most closely related, and we're going to see which ones those are. And how can you tell? You always look first at the genus. The genus here are these three right here. Now you can see that the two here who are linked, they both have the same genus. Now, but they have different species. As you can see, the next name here, which is the species, are different. And you can see that they are considered as two different types of species, even though they have the same genus. And here, right here, you can see that it's completely have a different genus and species. But if you're just looking from the picture itself, right? If you just look at these guys like this, you'll say, wait, they all look the same, but that's not true. It's because we're looking specifically in every part of them, every trait they have, and every behavior, every structure. So if they are located in the same genus, they're most likely to be related, even though they might be in different species. So always look first at the genus and then the species. And they should have the same, if they have the same species, that means they're the same type. So genus is where they can get the closest related. So looking for the example here, looking for which ones are most closely related. You can see that all of them, right, they all have canis right here. But then the next thing that they have most common is lupus and lupus, but this one doesn't have it. So most likely here that these two are more related than the other one. And then the last one, which is the species, that's what makes the three of them different from each other. So it's this whole binomial name, it's all about being how related they are to know each other. That's basically it. Now, just going back here, just myself, this is the kind of questions that can come out in multiple choice. So always look for the genus first. If the genus are the same, that means they're related, okay? Now, other types of classifications can happen. They can check for the way they feed, the places where they're living, what do they do during the day or the night, what kind of risk they have, like are they harmless or poisonous, and of course, if their anatomy. These are other ways that we kind of name the organisms. We're looking at all these traits in order to classify them. So in today's world, because of DNA, of course, a lot of the groups have been reclassified because we were just looking from the outside. But now that we know the DNA and we can compare the DNA, we can put them in a better position according to how close their DNA looks to each other. So it's not just about the naked eye. Now we can compare the DNA, and that means we reclassify so many things. So for example, an old classification would be the astrocordophilia, and now because of DNA that we can actually figure out the sequence of the A, T, C, and G, it completely got a new classification. So a lot of organisms that were classified, let's say 50, 60 years ago, now has changed because we have better technology to actually read the DNA sequence. So let's focus now on the taxonomy. It is a long topic. It has a lot of information to memorize. So this video, I'm just going to go over it, but it's important that you do many white card notes, writing down the traits on one side, getting pictures, or making projects in which you go for each one of these sections. So let's go forward. So here we have the level classification. We have the domain, and after domain, we have the kingdom. The kingdom goes to the phylum, the class, order, family, genus, species. You have to know the order. Now, as you're going up here, the taxa includes more species and share fewer features. That means up here, you have all the species in there, but they might just have, for instance, one or two traits in common. Everything else is different. But as you go down here, when you get to the down, they have more features, but they have less species included. So here on, for example, genus, they have a lot more traits in common, right? But of course, there's going to be less organisms in there, right? So maybe here you have 1 million of organisms, but with just one or two traits that the same. And here you might just have 10 organisms with 1 million examples of same trait. So that's how it works. And you need to know this because this might come out as a question in the IB exam. So once again, when we look at the domain has all the species, that means the large amount of organisms, but when it comes to their traits, their similarity is very low. When you get to the genus and species, here they have almost a lot of things in common, a lot of traits in common, but you have less numbers of organisms in there. So here is basically showing you an example. So we have here in the kingdom, you can see you have so many different types of animals, right? And then as you keep going down, you can see the phylums, they start to reduce. Why? Because only these animals include in phylum Chordata, so the butterfly is not there. When you get to the class, it gets even to shrink even more. And when it gets to the order, you get less animals in there, in the family less animals, genus less animals, and then species you're going to have just one type. So you can see that the number of organisms is decreasing, right? As you get down to the taxa. But if you go up, you have more organism. Now, also, as you go down, there is more traits in common. That's the most important part. Here you have a very little amount of traits. So this is very little, one or less. Now, let's talk about each one of them. In the domain, there's 3 domains that you must know. The archaea, eubacteria, and eukaryotes. Now, virus here, we are not considered as not living organisms, so we don't even put virus in this situation. Now, when we talk about these domains, they're divided into 7 taxas. And that means kingdom, phylum, class, order, family, genus, species. You have to memorize this order. So let's talk about each one of these domains. Now, archaeans, they are basically single-celled, and they can live in so many different areas, from hot springs to mammal guts, and they can live in very extreme places, such as, for example, in very salt places or very heat-loving places or where there's a lot of methane. That's why we have the methanophiles, the halophiles, and the thermophiles. Now, eubacteria in this case, they are single-cell organisms, and you can see it's the same thing as the archaeans. They can find in your gut, in your intestines, and they might cause infections. And here are some of the pictures of them. Then you have the eukaryotes, which is us. They are also known as sickle cells, but they can go from the smallest thing like yeast to large organisms such as elephants. They do have membrane-bound or nucleus, as well as membrane-bound organelles such as mitochondria and chloroplast, where the other 2 did not have. So what makes eukaryotes separate from the bacteria and archaeans is the fact that they have nucleus and they have mitochondria and chloroplast going on. Now here is the other domains that we have, the other kingdoms, but we don't have to know all of them. So at least you do need to know the 3 domains. When it comes to kingdom, you only need to know the Animalia and the Plantae, even though there's the fungi, the protistas, and Monera. We don't need to know these guys at all. So we're just going to focus on the Animalia and Plantae for IB. So let's look at the plant phyla, right? In this case, the kingdom Plantae, we have many things in common. They have photosynthesis, chlorophyll, cellulose in their walls, and store starch. These are very common things they should know by now. So everything is based on the external observation when it comes to the phylum of plants. So let's look at their external side. So we have 4 phylums that you must know for the plants. We have Bryophyta, Felicophyta, Conifophyta, and Angiospermophyta. As you can see, moss is an example of Bryophyta, ferns for the Felice, conifer for the coniferous, and flowering plants for the Angiospermophyta. So you have to know examples for each. Now we're going to go through each one of them. And this chart here, which comes from BioNinja, is a perfect chart because it kind of summarizes everything for each one of them and even gives you pictures. Okay, so the most important thing is looking at their structure, their vascularization. That means, do they have xylem and phloem? What is the kind of way they reproduce? Other features and examples. You need to know all of these. So we have here the 4 types of phylums. Now, looking at all of these, then we look at the structures. Bryophytes, they don't have leaves, right? Not even roots or stem, which that means they don't have any kind of like a tube, like the xylem and the phloem taking the food up or the water up. They only have spores as reproductions. They are anchored by rhizoids, which is like kind of like a fungus coming out, and these are mosses. The phyllosophites, they do have leaves, roots, and stems. They do have vascularization. That means they have the xylem and the phloem. They do have spores. The leaves are pinnate and they're fern. Conifers, they have leaves and roots and stems. They have the xylem and the phloem. That means they have vascularization. In this case, they have seeds. Okay, but these are naked seeds. They have wood stems and conifers. Angiosperms, they have leaves, roots, and stem. They have vascularizations. They have seeds, but these seeds are closed. They're closed inside the fruit. And they have flowers and fruits. And, of course, examples of flowers. So we have here examples like moss, fern, pine tree, and sunflower. You have to know examples for each one of them. So I'm going to go very quickly on these guys, but it's important that you stop each slide, write it down. As many times as you write it down, the more you will remember for each one of these. Also, very important, know examples. So right here, we have mosses, liveworts, and hornworts. These are examples of bryophyta. They are very much small plants, which that means they don't have the vascular tissue. That means they don't have xylem and phloem. That means they're like in the soil, they're so tiny like this that you don't need to have a tube that takes, for example, the water here, right, to go all the way up here. So you don't need to have some tube to bring the water up here because they're so tiny in the soil, which they also don't have roots, stems, or leaves. So they're very tiny and they're very spiral and they don't even have cuticle. And they're all sending spores. So then when it comes to reproductive system, it's all about releasing spores. So maximum you need to know about them is that they don't have vascular tissues, they don't have roots and stems or leaves, and they're made of spores. That's the max, and of course the examples that we have here. Next one is Phyllosynophyta. For these, think of phyli with F as in ferns, right? Now they do have the vascular, they do have all leaves, and of course they release spores just like the bryophyta they do. And examples we have is fern. When it comes to coniferous phytes, think of conifers. Conifers, they're the same thing, which is pines. You can think of this right here, which is basically a cone. Now, they have wooden trunks, right? They do have long thin needles, and they do produce seeds that are found in the cones. But again, these seeds are naked, and they do have a vascular system because these are like those big, huge trees, right? So they need the vascular system to take, for example, the water to go all the way up here to the leaf. So they need the vascular system. However, the seeds here, they are known as naked seeds. And then you have the angio. When we talk about the angiospermophyta, these ones are basically all the things the flower. That's the only phylum that has the flower. So that's what makes them different from the others. The flowers and fruits and the seeds are inside, so they're enclosed. They also have the vascular bundles. That means they do have the phylum and phloem. They have many leaves. The leaves can be big and small, depends on the situation, and they have wax cuticles. So for angiosperm, the most important thing is about the flower and the fruits. That's what makes a difference from all the other 3 phylums that you need to know. So here is again a very nice chart that I've created so you to summarize the phylums of the plants. So you have here on the side, you have all the 4 types of phylum. Now you can see gymnospermatophyte is the same thing as the coniferous, so it's the same word, it's just different written. So when it comes to vascular bundles, do they have xylems? Bryophyta, which is the small one, is the only one that doesn't have, the rest has it. And then when it comes to seed, bryophyta and phyllocytes, they don't have it. The gymnosophyte, they do, but it's naked, while the seed is closed because it's inside the fruit. When it comes to fruits, none of them has it, but the angel has it. When it comes to flower, none of them has it, but the angel does. So you can see that the angiospermatophyte has all of these, and you can see as you go up, it reduces. So this is a perfect chart summary to memorize to make sure you know the difference between the 4 phylums of the plants. Now we're moving to the animal phylum. So there's 7 of them that you must know. Now, looking from here, very careful, there is about these guys here, none of them are vertebrates. They are all considered as invertebrates. They don't have a backbone at all. So the only one that is a vertebrate is the chordata. So for peripheral, we have sponges. For Cnidaria, we have jellyfish. For the platymelon helminths, we have flatworms. For Annelida, we have segmented worms. Molluscas, we have snails, clams, octopuses. And Arthropoda, we have insects, spiders, and crustaceans. You have to know these names, and you have to know examples. So here we have a very summarized examples and the names that you should know it by heart. Here is the anaconda. So like I said, In the invertebrates, we have Porifera, Cnidarian, Phalanophytes, Annelida, Mollusca, and Arthropoda. All of these guys do not have a backbone. And you can see in the corner here, you have the vertebrates. In this case, you have the chordata written right here. So again, make sure you also know examples. And that's why I'm putting the examples here. It's for you to get exposed to the pictures and see what they look like, because these pictures falls in IB exams. They put the picture and then they say, hey, to which phylum does this thing fall to? So let's look at each one of them. First, the sponge. First, they are sessile. The word sessile means they don't move. Okay, they don't move. They're just standing there, stuck in a place. And they don't have a mouth or digestive tract. They have no muscles or nerves. They have no organs. So basically, they're just standing as sponge, doing nothing. When it comes to cnidarians, in this case, you can see the examples here on the bottom, but they also are stinging cells, right? They also don't move. Others may swim free, but they still, most of them are very much not moving. So they catch their foods by using tentacles, as you can see the tentacles here. And they have a gastric pouch with only one opening. That means whatever comes in comes out from the same opening. Then you have the platymelophytes. In this case, they are one body cavity. When it says one body cavity, that means the gut with an opening for food to enter and a waste to exit. So, for example, they are known mostly as parasites, and the perfect one is the tapeworm. Tapeworm is a perfect one for this phylum. And it's flat shaped and it's not segmented. Segmented will be like, imagine this is the worm, it would have been like this, cut into pieces. That's considered segmented, but it is not segmented whatsoever. So, and it's very flat, wide, to allow the exchanges of gases in and out much faster. Then you have the annelida. Annelida here is the one that has the segmentation. So you can see right here, these lines coming out, these are segmented. So it can be earthworm, leeches, and so forth. They're divided by rings, one mouth at one end and opening on the other end. So they do have a gastric that has an mouth and an anus. Mollusca. These guys usually have shell with calcium, okay? And they're one-way digestive system, which that means they have the mouth and the anus, and they're not segmented. So again, you have to know these main things. Things that are bold here are the ones that mostly fall in IB. Arthropoda, they have a hard exoskeleton composed of chitin here. They have body segmented, which is jointed together, and they have jointed appendage, which means limbs to each of the segments. So these are the main things, and this can be crabs, shrimps, insects, spiders, Scorpios. So make sure that you go through these slides again many times as you can and write on white cards, because there's no way that you just look at them once, as I mentioned, that you will memorize. So the more you draw them, the more you write them on the paper, white cards, make a huge project, make a website in which you're putting these animals and these plants all together with their traits. This way you'll be able to memorize. So here it's like a summary of all the 6 ones without the chordata, of course, of the phylums of the animal. And here it has all the things that you need to know for each one of them. So you can pause this and put this in a white card for each one of them. Okay, now just some of the words here to make sure that you know them. Like the word symmetry here, it just means that each side of the body is exactly the same, but this porifera doesn't have it. Another word that my students might have a problem is the word segmented. As we said, segmented, here it means that it has lines like these. Bilateral, when it says the word bilateral, that means there's 2 sides that are equal. So there's only 2 sides that are equal. Molluscas, let's talk about here. Segmentation not visible, mouth is anus, this is easy to know. Radial symmetry in this case is, for example, starfish, or that means, like, for example, you have the star here, right? That's radial. So you can see that these 5 sections, 1, 2, 3, 4, 5, they are exactly the same. Stingy cells, they basically just throw poisons. Exoskeleton is basically the skeleton on the outside. Segmented, we mentioned here. Joint appendages, we're talking about the legs being joined. Flat bodies, and again, the rest is all the same. So here is just a very perfect example. This right here, take a picture, write them down, Put it in your wall every day in the morning, look at it again. So if you're looking every day, just once, just read it. I guarantee you, if you do this, you put this picture on the wall, every day you wake up, just read it, and then go. That's reading will take you 30 seconds, and then the next day you wake up and just read it. And you do this for 2 weeks. I guarantee by the third week, you're already gonna know most of the information because by repetition, seeing it every day, just for that 30 seconds, it will get into your brain. Here's another chart, same story. This chart is showing different types of things such as the symmetry, digestive tract, segmentations, and other features. So you need to know for them. These are the most common things that comes in IB. And it shows here if they're radial or bilateral, if they have the mouth and anus or none. Here is talking about if they're segmented or not. And there's some extra features they need to know. So this is also a perfect chart that has, you can read it and you have to memorize. I'm not going to read it through because it's so obvious. You can just put a pause and write these down because these are the most traits that I see in IB exams as multiple choice. Now we get to chordata, which is also animal phylum, but this is separated from the rest because this one has the notochord, which this notochord eventually will become a backbone. So like birds, mammals, amphibians, reptiles, fish, sharks, all of these go from cartilages to become bones. So they are known as vertebrates. Okay? So here is one of the requirements by IB is that you need to know one animal and be able to tell his names from the kingdom all the way to the species. So you can see the kingdom is Animalia, the phylum is Chordata, the class is Mammalia, order is Carnivora, family is Felidae, genus is Panthera, and species Panthropods. Literally, you have to memorize this. Now, I put the panther because I love the panther, but you can choose any animal you want, and you will have to memorize their names on each one of the taxas. You have to know, not only with the animal, but you also have to do with the plant. So let's continue here. So now that we know that there's a kingdom Animalia, and the phylum is located inside the Animalia. Now, inside the phylum Chordata, there's a class, and there's 5 of them. Fish, amphibians, reptiles, birds, and mammal. Now, if you don't know these, then you probably are an alien because these are the most common things that you find in the zoo. So, first the fish, you can see that they possess the gills. That's one of the things that comes in IB. They also have fins. Okay, these are the most common things that I see fall in the IB exams is they have gills and fins, okay? And scales. Now, amphibians, one of the most things I see about them is that they have gills, okay? And they develop lungs after going from they always go from the gills here and to become lungs and when they get to be adult. So they have eggs that don't have a membrane around the embryo, okay? They are also known as cold-blooded, okay? And they also have moisture skin. I don't think it's written here, but they have a moisture skins, the amphibians. And then next is the reptiles. When it comes to reptiles, they are ectothermic too. They have scale. They are seen in birds and mammals, but what is seen also in mammals and birds is the amniotic eggs, which that means it's a membrane that is surrounding the baby, right, the embryo. So the reptiles, they do have amniotic eggs, and you can see the options of the pictures. Then we have the birds. They mostly have feathers and lay eggs. They have hollow bones. They peak with no teeth. Okay, these are the things that you need to know that usually falls, again, like an IB, okay? And then the mammals, which are us, they have hair. Hair is one of the traits I see a lot in IB. They produce milk. They are endothermic. That means they regulate their temperature. They maintain their internal temperature always fixed. This is pretty much it. They have 4 limbs, most of them. So here, again, it's a picture from BioNinja, which is perfect because here they cover the body. What are the things that covers is the scale, is the skin, is the feathers, is hair. Then what kind of reproduction they have, what kind of way they breathe, the temperature that they have, and other features. So this chart here is a perfect chart by BioNinja, and I will take this picture, put it in a wall, and watch it every day for 30 seconds. I guarantee you by the end of 2 weeks, you have mastered this very much. Here are the 2 examples that the students can use. So if you have to memorize, because that's one of the requirements, you have to memorize one for the animal and one for the plant. So here we have the humans, kingdom Animalia, Chordata, Mammalia, Primate, Hominidae, Homo sapiens. For the garden pea, we have Plantae, Angiospermophyta, Dactylognoa, Rosales, Papilionaceae, Piscium and Sativum. Now, you don't have to memorize these twos. You have the choice of choosing which animal and which plant you want to memorize, going from the kingdom all the way to the species. That is up to you. Has it fall? I have seen it once. They asking about the human one, but I haven't seen them still yet, and I be asking to give out these ones for the plants. So now let's talk about the dichotomous key. Now, dichotomous key is a way for us, let's just say, we went to the forest, and by chance, they found this new organism. They have never seen this organism before at all. And they're like, oh shoot, we have to figure out where does this organism belong, to which group, to which kingdom, or which domain, which phylum, which class, which species we're going to put. For them to do that, the first thing they have to do is to figure out, hey, what are the traits? What are the behaviors? How do they feed? So the most important thing about the dichotomous key is to place them in the right taxa. So like I said, the first thing when they see the species is that they're going to look for their traits. Like, what are their traits? How do they feed? How do they sleep? What kind of blood do they have, right? Right? Do they have bones? Do they flower? Like, these are many questions that we have to ask. So they observe this for months, the species. And when they do this, then they go to this dichotomous key, which is a huge set of organization in which they try to figure out through a yes and no going on like this. And they find the group in which that species will fall to, or maybe they create a new group. So we're going to look at an example of what a dichotomous key looks like. Here we have 4 organisms, right? Let's just say that we don't know. We have never seen these organisms. So I'm going to pick, let's say I'm going to pick right here, in this case, the turtle right here, right? I just saw this turtle. Now I take it to the book of dichotomous key and look at it. So here is what a dichotomous looks like. You have number 1, no differentiated tissues, no symmetry, no identified organs. If this animal, turtle, had these traits, then he would belong to the group of Porifera. But he does have differentiated tissues and organs, so he goes to number 2. So he jumps to this next one. And number 2, stinging cell presence can show radiosymmetry. If he has those traits, then he's Cnidaria, but he doesn't have radiosymmetry, no even does stinging cells. So because he has no stinging cells, he moves to 3. So he jumps to 3. Now, he has 2-way distract and bilateral symmetry. If he has that, then he's a platy, but he's not. He has a 1-way, which is a mouth and an anus, so he goes to number 4. So he gets to 4. Does not possess a notochord at any time, he goes to 5. If he does possess a notochord at some stage, he goes to 7. Well, here's where you need to know about the turtle. So they do have it because they're coming from the chordata, so you jump all the way to 7. You don't skip 5 and 6. 4 limbs present with articulated digits, you go to 8, or limbs present but they do not have digits. In this case, they do have digits, so they go to number 8. So when you get to number 8, does not produce a minion, that would be amphibians. Can't produce an aminion, they go to 9. So this is where you have to figure out if they are or not. So because they produce a minion, they go to number 9. So number 9, presence of hair on the body can make milk. Well, that's not true. But the absence of hair cannot make milk. That is true. They go to 10. Covered with feathers. That's not true. They're covered with scales. And there you go. You put the turtles in the reptile. That's how you go through the dichotomous key for the turtle. And you can do the same thing if you want for all the other animals. You just go through this process. If you notice in this process, it's all about, do you have this or do you have that? So we have here numbers being placed, right? And in these numbers, there's always an A and a B, an A and a B. And sometimes you are talking on through A, B, the same type of trait. So for example, here, do not possess a notochord, or it does possess a notochord. So you're talking about that one trait. Sometimes you can put 2 traits. And then here, you usually will put the phylums that they fall into, or the numbers that you want them to go for next. So this is what the dichotomous key usually looks like. And like I said, you can do this for all these species, like we did for the turtle. You can do for the moth, you can do for the jelly, as well as snail. You can do for all of them in practice. So now one of the requirements IB is that you'll be able to create a dichotomous key. This is a dichotomous key for vertebrates, right? So you can see here, if you have fur, you're mammals. If you have no fur, then you go to next. Do you have feathers or not? If you have, you're a bird. If you don't, then you go next. External or internal fertilization. If it's internal, you're reptiles. If you're external, next. Gills as adult fish, no gills as amphibians. If I had to write this as a dichotomous key, I will start on top. So if it has fur, then you are a mammal. I'm just going to put mam here. Now, if you have no fur, right, then you go to number 2. So it goes to number 2. Number 2 is the next thing, which is the feathers. So if you have feather, then you are bird, right? And if you have no feather, then you go to number 3. So go to number 3. Number 3, if you have internal fertilization, then you are reptile. And if you have external, then you go to number 4. Then number 4, if you have gills, right, you are fish. And you have no gill, then you are amphibian. So this is how you create a dichotomous key. You can also do the same thing with the other phylums between the invertebrates. So the same thing, usually the number 1 will be, do you have asymmetrical, then you peripheral. If not, if you're symmetrical, then you go to number 2. Number 2 is if you're bilateral, you go to number 3. If you're radial, you're cnidarian. And then if you have no anus, you're platy. If you do have anus, you go to number, this will be number 3, I think. So this means you go to number 4. If you have no visible segmentations, you're molluscus. If you have visible segmentation, go to number 5. If you have exoskeleton, you're arthropoda. If you don't have exoskeleton, you're Annelida. So that means you can build up the dichotomous key. Here you can also do with the plants. So for example, if first you're going to say if you have no vascularization, you're Bryophyta. If you do, then you go to number 2. If you have no seeds, you're Phylum. If you have seeds, go to number 3. If you have no flowers, you're coniferous. If you have flowers, you're Angiospermae. So you can see it's very easy to build once you have this key right here. So this is pretty much how the dichotomous key is built and how you can read it. Here is a study guide with all the questions for 5.3. If you know how to answer these ones, then you have really nailed 5.3. Here's more questions that you need to be able to do. And that's pretty much it for the section on 5.3.